Mr. Keith.
My lady, the first witness today is Professor Sir Nicholas White. Could he be sworn, please?
I swear by Almighty God that the evidence I shall give shall be the truth, the whole truth, and nothing but the truth.
Professor, could you commence your evidence, please, by giving us your full name?
Nicholas John White.
Thank you very much, and thank you for attending today, of course, and thank you also for your provision of your expert report, dated the 15th of December 2024. Just in terms of the formalities relating to that report, it is obviously your own work, where you provided, for the purposes of writing that report, a number of documents, quite a lot of material, consisting in the main of witness statements submitted to this inquiry by witnesses, as well as some, but not, I think, all of the expert reports that the inquiry had commissioned.
I was.
Commencing, please, with your qualifications, you are Professor of Tropical Medicine at Oxford University and at Mahidol University in Bangkok. You've been for many years a consultant physician at the John Radcliffe Hospital in Oxford. You've chaired for some time the Wellcome Southeast Asian Research Units. They're in Thailand and Vietnam, is that right?
Yes.
And for many years, your specialty, the area of research, has been on infectious diseases and in particular the assessment of therapeutic responses.
Yes, that's correct.
In the course of the COVID-19 pandemic, were you in fact directly concerned in one of the major trials that was undertaken at that time?
Yes.
Was that the Kopkov trial? Well, we'll come to that later. And therefore, you were directly concerned with an important piece of pharmacometric assessment.
Yes.
You also chair, is this right, the Scientific Advisory Committee of the Drugs for Neglected Diseases Initiative. What is that?
It's a non-governmental group based in Geneva, founded with the money from the MSF Nobel Prize, which develops drugs for diseases that predominantly affect low-resource settings.
Thank you. Happily for you, Professor, much of your report has been traversed through the evidence that my lady's already heard in the course of this module. And therefore, by the happy coincidence that you're almost the last witness in this module, much of your report won't need to be re-examined. But I'd like you, please, to start by explaining in the context of trying to respond to a pathogenic outbreak, a disease, why it's so important to have drugs or therapeutics available, whether or not there is a vaccine available.
So we need, in the context of an outbreak or a pandemic, we clearly want to stop people dying and suffering. And the way we do that in the longer term is by developing a vaccine, but that takes time. So before a vaccine is generally available, and for the many people who can't, or some who won't take that vaccine, we need to have medicines that will both, if possible, prevent and also to treat, with the objective, as I said, of reducing suffering and death.
From your report, it's obvious that there are a number of different types of medicines or therapeutics, from small molecule drugs to what are known as neutralizing monoclonal antibodies. Is it of benefit, in very general terms, to have as wide a variety of types of drug or medicine as possible?
Initially, yes, because we don't know what we are facing. Ultimately, you would like to be able to develop a highly potent, highly cost-effective, safe, well-tolerated medicine. but at the beginning you don't know. So yes, you need to have a large armamentarium from which to choose your best weapons.
Does the time that is required to identify, develop, and make available a drug vary between different types of drug?
Yes, it does. It depends on many factors, but yes, it varies.
In the course of responding to the COVID-19 pandemic, it's self-evident that some drugs which were already in production and available and for which authorization had been given were repurposed to face the exigencies of the COVID-19 pandemic. Other drugs had to be developed from scratch. Presumably, those repurposed drugs are able to be brought to the marketplace and to be made available much more speedily than new drugs. That's self-evident, isn't it?
Yes, that's correct.
And therefore, for the purposes of that latter category, new drugs, you've got to have up and running, have you not, an efficient, rapid, and well-managed process by which those drugs can be identified, trialled, and ultimately authorized.
Yes, because the repurposed drugs in general will not be as good as the bespoke drugs that you develop specifically for the infection.
And why is that? Is that because if they had been suitable for the particular pathogenic outbreak, they would have been already authorized for that particular purpose?
Yes.
And in the course of this pandemic, of course, one of the greatest successes was dexamethasone. Was that a repurposed drug?
Yes, dexamethasone is a drug, it's a corticosteroid, a drug that's widely used in medicine. And it was not really expected to be very effective in COVID.
Given that one of the most successful therapeutics was in fact a repurposed drug, why is it necessary to still keep on pushing for the identification and production and manufacture of new therapeutics?
So dexamethasone was an anti-inflammatory drug. It's not a drug specifically for infections. It's a drug to stop the harsh effects of infection on the body. But you need specific drugs to treat infections. We call them antibiotics, antibacterials, antivirals, anti-whatever the nasty pathogen is. And those ones you need to develop. If you haven't got one already, we need to develop it specifically. And we did not have any drugs available specifically for coronaviruses.
You say, I won't take you to it, but you say at page 84 of your report, the more severe the illness is, the broader the potential use of antiviral medicines. What did you mean by that?
In a potentially lethal pandemic, your primary objective initially is to save lives. And so anything that works is to be welcomed, but if it doesn't work very well, clearly you're going to have to try and develop something which is more effective. So initially we had no idea what would work or what didn't work. The first studies showed that a lot of the things we thought might work, the repurposed drugs, didn't. And then we were very gratified. In fact, the most important outcome, I think, of all clinical research in COVID was the discovery that steroids work. That was a surprise, but it saved lots and lots of lives. But then we still had to address the problem of the infection itself. So we had to get drugs that worked to stop that infection that we could use in the community, we could use to prevent the infection, and we could save lives in hospital.
And was dexamethasone, in fact, a corticosteroid?
Dexamethasone is a potent corticosteroid.
So just on this issue of, or in very broad terms, of the benefit of therapeutics, it's important to make sure that you can identify, develop, and make available drugs that reduce not just hospitalization and death at the topper end of the terrible consequences of a pathogenic outbreak, but that you can also develop drugs and make them available that reduce the symptoms of the disease and the general levels of illness that they bring about.
Yes, in general, yes. So, but if I may amplify that a little,
please.
We try to develop vaccines to stop people getting ill at all. And we have tried to persuade people, we provide the evidence that these work. if people do get ill for whatever reason, either they didn't get the vaccine or it didn't work, we still need to treat those people. Ideally, from a public health perspective, we want people to not go to hospital and die. So if we can treat people early in the community, that's better than later on in the hospital when they may become sick and die. But of course, you have a trade-off there, because and so that's why we have to identify who needs it most. Can we afford to give it to those people? Is it safe and well tolerated, and so on? So in the end, you have to make some choices. For drugs that are very safe and very well tolerated and very inexpensive and affordable, we deploy them more widely. And for drugs which are not so available, more toxic and so on, we are more restricted in who we advise should get them. And we have this debate commonly about antibiotics.
Still on the subject of the potential benefit of therapeutics, the great benefit and the great purpose of the use of dexamethasone was recognized, I think, in the summer of 2020. Vaccines were not, of course, made available until the winter, till December of 2020 and then January 2021. But had new therapeutics been identified, trialed and made available in the summer of 2020, particularly antivirals, would that have had a dramatic impact upon the course of the pandemic and the response in terms of societal illness and the consequences?
I think so.
And is that what the aim has to be in any future
Yes.
pandemic, is to make sure that you've got that option of therapeutics being available pending the development or perhaps in the complete absence of a vaccine?
Yes, COVID killed 7 million people around the world. That's far too many. We don't want that to happen again.
Turning to the issue of the trial process, we've heard a great deal of evidence about the way in which, in particular, the phase 2 clinical trial process operated in the United Kingdom. It is clear from your report that there was a great deal of funding available for clinical trials. We've seen some evidence to suggest that the clinical research network of the National Institute of Health, now the Health and Care Research, NIHR, received, I think, over 1,600 applications for trials. 101 of them were badged with urgent public health approval. And there were, I think, over 1 million participants. Is that right? Some 1.3 million participants. So it's obvious that a great deal of attention was paid to the trial process, and it was a massive undertaking. At the same time, Professor, the inquiries received some quite critical evidence that's quite critical of, in particular, the phase 2 therapeutic process from Sir Jeremy Farrar, from Eddie Gray, and Professor Sir John Bell in particular. At the heart of any clinical trial process at the phase 2 stage, are there quite considerable difficulties associated with identifying the standard that must be reached, what that phase 2 trial is trying to achieve?
Yes. So let me say that overall, if we look at medical research in COVID, the UK was very strong in clinical research, and perhaps the weakest part of that impressive portfolio was phase 2. So phase 2 is that transition when you've got a drug, a new drug, or even an old drug at a different dose. Your first attempt to try and see does it work in people or not. And for that, you need a way of measuring that. And unfortunately, we didn't have a good way of measuring it. And what people did was a large number of small and unfortunately inconsequential trials, which could never have answered, did the drug work or not? We needed a better method, and that was not developed.
And just pausing there, it's self-evident that the more people you have in a trial, the better, the wider the range of results, of course, and the firmer the conclusions are that can be drawn from it.
Yes, this is a very important point. When you have a very large benefit, you do not need many people to be studied to show that very large benefit. But when you have a relatively small benefit, for example, a reduction in the death by 20%, you need very many people to show that with confidence. So yes, large trials, in general, trials are too small. Very seldom are they too big.
We'll come back to the question of why and what can be done about ensuring that there aren't so many underpowered, that is to say, small trials next time in a moment. I enter into the next topic with some degree of trepidation, Professor, but in the course of phase 2 trials, there are two particular assessments which are required to be carried out, are there not? One is that of pharmacokinetics, that is to say, assessing the concentration in the body over time of the amount of drug which is left in it, in the body, and secondly, the issue of pharmacometrics, which is trying to assess how quickly the infection in the body is resolved by the receipt of the drug. Is that broadly right?
Yes, so very briefly, pharmacokinetics is what your body does to the drugs, how high the levels that you get in the different parts of the body. Pharmacodynamics is what happens to the infection in this case, how quickly it goes away, how quickly it's killed. And melding those together is called pharmacometrics. It's a rather complicated terminology, but it's the science of trying to understand how much drug do I need to give. First of all, does it work? How much do I need to give?
And a phase 2 trial in general terms will only be effective or useful if it can answer those questions.
Yes.
Are there standards, technical standards in place, as far as you understand, to ease the path of the scientists and the clinicians and the researchers in answering those questions?
I think this is one of the weakest parts of drug development. It requires innovation and, in general, the regulatory authorities who ultimately make the decision whether a drug should be registered or not are fairly conservative, both on this side of the Atlantic and the other side.
So is one of the recommendations that you invite my lady to consider the production or the call for clearer technical standards in order that everybody may better assess the worth or the efficacy, the effectiveness of phase 2 trials?
Yes, standards and methodologies.
That's on the technical side. Then in terms of the administration of this process, there are obviously, we've heard from the evidence, a number of moving parts from the setting up of the trials, the funding of the trials, the regulation and the administration surrounding the trials, and of course, recruitment. Is it your assessment that there needs to be a higher degree of coordination on the administrative side? That is to say, a better management of the funding, the setting up, and the oversight of the phase 2 trial process?
In simple terms, yes. We have to have some sympathy at the beginning of this dreadful pandemic for the organisation or disorganisation. But ultimately, I think it should have coalesced into something simpler. We had probably too many committees, but yes, overall, my short answer is yes.
You're aware that in her Rule 9 witness statement, Professor Charlotte Dean of UKRI, UK Research and Innovate, is it?
Probably.
The acronyms have finally on the last day defeated me. UKRI, anyway, the very significant funding body. She has suggested... I'm sorry, my lady?
Research and innovation.
Research and innovation, there we are, thank you very much. She has suggested in her witness statement that there should be a UK-wide structure to select potential interventions, to direct resources and to coordinate with regulators the rapid setting up of trials. Would you agree?
In general, yes.
As it happened, there were, though, nevertheless, some extremely impressive and effective phase 2 trials. Is that right?
There were some.
There were some.
There were some, and there were a lot of ones which were not. And I'm afraid the ones that were not were the majority wasted resources and effort.
And so that we can be clear, not to damn the very effective phase 2 trials with that observation, Professor. Agile is a trial of which we've heard much, I think, at the University of Liverpool, which was one of the very effective phase 2 trials.
Yes.
Accord is another trial which produced very useful and effective results. If there were a better UK-wide structure for the administration, funding and oversight of particularly phase 2 trials, do you think there would be a need for the amazing proliferation of panels and review groups that appear to have been in place during the course of the pandemic, such as the UK COVID Therapeutics Advisory Panel, the Therapeutic Subcommittee of Nerve Tag, Rapid C19, the Neutralizing Monoclonal Antibodies, Antivirals Independent Advisory Group, and the Prophylactic Oversight Group?
The short answer is I think that could have been rationalized, but I understand how it evolved.
Turning to the important issue of diversity, diversity may be measured, of course, in a number of different ways, but perhaps two of the most important ways are ethnicity and age. How important is it that all clinical trials have a sufficient depth and breadth of diversity?
Yeah, this is a very important point. I think it depends at which stage of the development we are. So it's not necessary at the beginning, in for example, the pharmacometric studies we've just discussed, to have a range of diversity, because these are small trials. Then, based on prior knowledge and understanding of the drugs, when you go to the definitive phase 3 trials, which are the ones which precede the regulatory approval, at that stage you do need to proactively try and engineer diversity. But I think there's an important point, if I may add, and that is that in the context of a rapidly evolving pandemic, which is killing lots of people, speed is paramount. And that speed benefits the diverse population. So getting to an answer more quickly is more important than having an engineered diversity which might slow down the recruitment to the trials.
Well, that's very clear. In general terms, but particularly by comparison to vaccines, do therapeutics have the same effect on all aspects or all parts of the population? So are there sectoral or subpopulation groups who may be more adversely affected by the receipt of a therapeutic, or who may not benefit to such a degree as other parts of the population?
Yes, there are well-described genetic and therefore ethnic differences in the way our bodies process drugs. And that we know a fair amount about, but we need to be aware of that with new drugs and to proactively try and investigate whether there are issues that might result ultimately in a different way of dosing, or some people not getting the drug, or not you recommending against giving the drug to a particular population. So yes, short answer, yes.
So diversity is vital not just for the purposes of promoting public confidence and reducing hesitancy in the receipt of therapeutics, but it's directly relevant to both safety and effectiveness.
Yes, in general, it's in general not a problem, but it can be an important problem, and therefore in developing new treatments one has to be aware of it.
I apologise, I forgot to ask you one final question in relation to phase 2 trials that I want to go back to. You'd mentioned you'd observed that there was, in your opinion, a large number of small or underpowered trials. Do you think that if there is a clearer technical standard, a better understanding of what threshold in terms of effectiveness and outcome phase 2 trials should be required to meet, and better administration of the overarching process, that there would be a reduction in the number of ultimately not very helpful, underpowered trials?
Short answer, yes, but I would like to emphasize that the method of doing the trial is critical. You can be very efficient in figuring out whether something works or doesn't using in a relatively small trial, but that was not the design that was employed in most trials. So it's the way you do the studies as well as the size of the studies.
And if the health authority, the DHSC, or the regulator, the MHRA, or its advisors, the CHM, set out more clearly how the trial should work and what is the ultimate outcome, do you think that problem would dissipate to some extent?
I think so. If I may give one specific example.
Please.
So with the monoclonal antibodies, there was, as I think you've all heard, some concerns about whether some should have been approved, some shouldn't have been approved, and so on. The overall problem was that the decisions on whether to deploy the monoclonal antibodies were based on tests in the lab. Those tests were never calibrated. So a particular value in the lab, it wasn't known what that meant. If the monoclonal was completely useless in the lab, it probably was completely useless in a person. But let's say the measure of how good it was had gone down tenfold. We had no idea what that meant in a person. You need to calibrate these things.
Was there another additional difficulty, which was that for many of the trials, the in vitro work was going on at a time, well, further towards the beginning of the pandemic. But by the time that certain drugs were authorized and then being utilized in the community, the context had changed violently, both by way of the success of the vaccine program, but also because there were variants circulating against which the drug might actually prove to be less effective.
Yes, so as we all know, the pandemic changed dramatically over a few years. So at the beginning, there were no drugs, nothing worked, we had no vaccine, and there were a lot of deaths. But very quickly, the vaccine substantially exceeded our expectations. I mean, WHO would have taken a 30% protective vaccine. We got much better vaccines than that, and that took the pressure off developing the drugs.
It's well established that there are two particular cohorts, children and pregnant women, who are, by and large, well, they're often excluded from clinical trials because investigators assess that it's simply not worth the risk. You don't know what the consequences might be of trialing unknown therapeutics on children and pregnant women. There is always the possibility, perhaps the probability, that the next pandemic, particularly if it's a flu pandemic, may hit children more or worse. How do we resolve that conundrum, which is that children, having been by and large excluded from trials, may nevertheless be the people for whom those trials will most benefit, or are required to benefit, I should say?
Yes, this is a very, very important point, and it is a dilemma. But in general, and I'll be very general, I think everyone, including the regulatory authorities, now accepts that we should much earlier, in the course of developing new therapies, evaluate them in children and have a plan for evaluating them in pregnant women, if that is the appropriate thing to do. So whilst I can't resolve the dilemma, I can just say that everyone accepts that it is a dilemma, and we need to move more quickly into these two very large and very critical subgroups of people who may disproportionately benefit.
Standing back, as you were asked to do in your report, and looking in an overall sense at the regulatory process that applied to the development and authorization of therapeutics in the United Kingdom, is it your view that in terms of output, and you refer particularly to the principal recovery and panoramic trials, the regulatory system worked well? It allowed the production of properly regulated and safe therapeutics.
Yes.
Do you have a view as to whether or not, in that difficult balance between imposing regulatory requirements, perhaps a certain degree of administrative bureaucracy, and speed of output, that the system worked well enough?
Reasonably well.
Professor, I detect a certain reticence on your part in answering that question. Do you, and the solution has presented itself, it may be thought, but do you say in your report that there is at least the impression that there was quite a significant degree of bureaucracy in the process, and the process was generally becoming quite a burdensome one in terms of the regulation of trials, but that as it happened since the pandemic, the legislative process for regulating clinical trials has been significantly amended by the introduction of new legislation, in fact, in December of last year?
Yes, and I hope that does facilitate the development of new medicines and vaccines.
Can we now turn, please, to the issue of Evusheld, which was, as we all now know, a monoclonal antibody, which was authorized, but which was never bought by way of advance purchase, and ultimately was not made generally available for use. It simply wasn't, a decision was taken that it wouldn't be made available. I'm not going to ask you any questions about the first stage of the process, that's to say the decision-making around the lack of an advance purchase in 2020. But I wanted to ask you, please, about the process that was applied towards the end of 2021 and into 2022, when there was a reassessment of whether or not it should be made available. In very general terms, a recommendation was made by, in particular, the Office of the Chief Medical Officer, in the form of the Deputy Chief Medical Officer, Professor Jonathan Van Tam, that the Evusheld therapeutic was required to be put through its paces in terms of an assessment by what was called the Rapid C19 Committee. There was a debate as to whether or not it should be assessed by another process called the Protect V process, and ultimately it was assessed by a body, a process operated by NICE, the National Institute for Clinical Excellence. It appears that all that took quite a long time, and by comparison to the assessment of the effectiveness of vaccines and other therapeutic drugs, it appears to have become somewhat bogged down. Why do you think that was?
I agree it was rather slow in comparison to other decision-making processes. I think the problem was, and I think some of the, this has been highlighted in some of the reports, was that there wasn't a way of extrapolating from the lab tests to the clinical use, as I just described. So it's a monoclonal so actually two monoclonal antibodies, and it was clear that as the virus was evolving, the lab tests were showing that the monoclonal antibodies were not as good as they had been before. But what did that mean in people? Nobody knew. And that uncertainty pervaded the decision-making process.
So coming back to the point you made earlier, was there a heightened requirement, something that wasn't in fact done, for there to be greater pharmacometric testing on the effectiveness of that combined monoclonal antibody?
Yes, and that was recognized by the panel, but none was commissioned or recommended.
Why do you think a decision was taken not to make it available
I think that
without those clinical tests having, or pharmacometric tests having been done?
I assume that the panels made a decision that these laboratory tests created sufficient uncertainty as to the value of the monoclonal antibody that it wasn't worth deploying it, I guess.
Might an alternative and perhaps better outcome have been to say, well, we won't make a decision about whether it should be made available until those pharmacometric tests have been done so that we can have a better understanding of the effectiveness of Evusheld?
Yes.
All right. And are you, insofar as one is ever critical in this scientific field, are you critical of that process in your report?
Yes.
By the time that decisions had to be taken as to whether or not Evusheld should be put through the rapid C19 process and ultimately the NICE process and a decision taken on benefit, had, as you were saying earlier, the context changed radically. So we're talking now about the beginning of 2022. Well, what had changed in terms of the use of a moderately effective therapeutic?
Well, everything was getting better. The vaccines were much better than we thought. People were acquiring natural immunity, and we saw the arrival now of bespoke, specific small molecule drugs, notably molnupiravir and then Paxlovid. So the need for other interventions was diminishing. However, had they been very, very good, I think they would. It would, as always, it's a balance between costs and risks and benefits and deployability.
Do we take it that if there had been those technical standards in play, of which you spoke earlier, if there had been a greater focus on pharmacometric testing, and if, as you say, speed and rapidity of outcome has to sometimes outweigh competing considerations, do you think if all that had been done, as you suggest it should generally be done, Evusheld, the outcome of Evusheld might have been different?
It might have been different.
And of course, Evusheld is a therapeutic which could have been of great assistance to, in particular, the immunosuppressed.
Yes, but we also have to acknowledge that that information, that calibration still hasn't been done, but it is becoming diminishing. Well, pretty confident it wouldn't work now, but was there a window of opportunity for Evusheld? Possibly.
Mr. Keith phrased his question in a particular way. Isn't it speculation to say it might? I mean, I appreciate it's conditional, might have, but because it's not been done, it's speculation, isn't it?
Yes.
And I think, to be fair, I've put it on the basis that we're concerned here with the processes and the systems. It's important to recognise that you've got to have a better system in place, and regardless of what the outcome might have been in an individual case. That then brings us on to the last specific discrete issue, which is hydroxychloroquine. Hydroxychloroquine was I believe, a therapeutic which was already licensed for some conditions. Is that right?
Yes, its older brother chloroquine was developed in 1945. Actually became the drug to which human beings have been most exposed. 300 metric tons were used every year to treat malaria. And then in the 1950s, but then more in the 1960s, it became clear that chloroquine or hydroxychloroquine could be used in rheumatological conditions like rheumatoid arthritis.
The issue of whether hydroxychloroquine should be authorised by way of repurposed use for COVID and whether it was beneficial at all became highly polarised, did it not, in
Yes.
the public domain? Why did that particular therapeutic, Professor, become so viciously polarised?
there was a subsequently retracted claim from a very prominent French, previously very eminent researcher, claiming that it worked in a very small trial. This was endorsed by heads of state on both sides of the Atlantic. This then made it politically polarized, and the scientific community became, I think, affected by this.
The study which raised the issue of whether or not hydroxychloroquine in fact would harm recipients or patients was published in an article. Was that article subsequently retracted when concerns came to light concerning the accuracy of the study?
Yes. If I may, just a little bit. So these drugs are very safe if you use them at the right doses, but they are dangerous if you use them at the wrong doses. So, and we knew that because of this extensive experience, both in malaria and in rheumatological conditions. But then suddenly, in May of 2020, this report appeared in the Lancet claiming that actually that hydroxychloroquine killed people and made the heart go too fast and that people died. But over 48 hours we investigated this and realized that this study could not have taken place as it had been described. And ultimately, it turns out that the data have never been provided. They couldn't have been provided. And the editor of the Lancet himself declared that they'd been fabricated. But the damage was substantial.
As a result of the publication, the MHRA, the UK regulator, got in touch with the investigators of, I think, the eight trials that were then going on concerning hydroxychloroquine, and posed questions of them as to what the meaning of the study was, of course, and no doubt asked for data. Did there come a time when, as a result of the article, recruitment in the vast majority of those trials was suspended, and of course the process came to a juddering halt?
Yes, so regulatory authorities across the world acted in a similar way. In the UK, the recovery trial was nearing the end of its enrolment with hydroxychloroquine and got a special dispensation from the MHRA. All other trials were stopped immediately. And then it took some time for them to restart. And by that time, the wave of cases had declined, so recruitment became difficult, and also the adverse publicity lingered. So many of the trials never restarted.
One of the trials, and you referred to it earlier, did restart, the Kopkov trial. That was the trial which you were directly associated with.
Yes.
But ultimately, on the 5th of June, the Commission on Human Medicines was in fact in session. It was informed through a press release from the recovery trial that the recovery trial itself had been stopped by its own investigators. Was that because nothing to do with any suggestion of harm? It just appeared that the data was beginning to indicate that the clinical benefit was less than perhaps had been hoped, and therefore was insufficient to justify the continuation of the trials?
Yes, it's an important point that recovery was a trial in severe disease. Kopkov was a prevention trial. So you have prevention, you have treatment of mild disease and treatment of severe disease. And it's not, you shouldn't extrapolate necessarily from one to the other. But the reason the recovery trial stopped was it was quite clear that hydroxychloroquine was not beneficial in the treatment of hospitalized patients.
But it might have been or could have been beneficial in the context of addressing symptoms and allowing people perhaps to return home or to recover more quickly in the community.
That would be speculation, but
but we'll never know.
We'll never know. But just to say that it's much easier to prevent a disease than it is to treat it. So drugs which are quite weak in treatment can still be useful in prevention.
And is it for all those reasons, the story of the suspension of the trials in hydroxychloroquine and the retraction of, or at least the difficulty in pursuing the trials at the time that the article was published, lead you to one of your recommendations, which is that in a quite a general sense, it's vital in the context of therapeutic clinical trials to protect the system, to protect that process from political or press speculation, and to allow the professionals, the scientists, the clinicians, the investigators to go about their work uninterrupted or unimpeded.
Yes.
Not quite sure how one could do that without impinging on the freedom of expression. How could one protect the system from media believing it's a legitimate interest upon which a public interest upon which they want to comment, and politicians the same? How would one protect the system?
Well, I think with the clinical trial, there are various ways in which a clinical trial can be obstructed or facilitated. So clearly a clinical trial involves recruitment of people. And it's really important that people have a fair chance to understand why, to give fully informed consent, as we say. So, of course, we can't protect people from learning all sorts of things, but what we should do is try and discourage politicians, journalists, from making claims which are not true and which might influence people adversely. I mean, this is of great contemporary interest across society at the moment. So I think that's the protection to maintain honesty and to try and facilitate the understanding of the science by the public. And then the other thing which the other organisations which bent under political pressure were the regulatory authorities themselves. And that shouldn't happen. I mean, the regulatory authorities should be absolutely independent. And they should not be pressurised, as they were, for example, across the Atlantic. The US FDA was definitely pressurised by government. And that should not be allowed to happen. I think that is something you can enforce. But I agree with you. You can't protect people from, well, in a free society, you can't protect people from access to information. But what you can try and do is ensure that lies are not told.
That brings us quite neatly back to the phase 2 trial process, to which you were speaking earlier, and the final remaining problem that arose in the course of the pandemic, which was, in a general sense, recruitment. And you referred to the problems of recruitment. Although overall there were a very large number of people who were recruited, and you've told us there were over 1 million people, a problem arose particularly towards the end of 2020 with reaching the right levels of recruitment for particular trials. There were trials going on and they were coming to a halt because they just couldn't recruit enough people. In the context of vaccines, there was a vaccine registration system which allowed members of the public to sign up to trials for vaccines. Do you think more needs to be done in terms of encouraging recruitment in the context of clinical therapeutic trials and or for setting up of a more formal system for recruitment through registration?
Yes, I think that can be done. It was done to a certain extent. The particularly favoured trials, particularly, for example, panoramic and principal trials, had extensive support via the National Health Service to encourage recruitment. And I think that could be, that process can certainly be strengthened.
And so turning then to your conclusions and your recommendations, we've covered many of them in the course of your evidence, but to summarise, you would endorse the strong rationale for having as effective and as a rapid a process of development for non-vaccines prophylactics and therapeutics. You would endorse the need for a better coordination, both by way of technical standards and administratively, of, in particular, phase 2 clinical trials. Thirdly, you make a broad appeal for better pharmacometric analysis to be carried out in the course of particularly phase 2 trials so as to avoid the sorts of problems that arose in relation to Evusheld?
If I may interrupt.
Yes, please.
Specifically to calibrate laboratory tests which are used to inform decision making.
Thank you. And finally, this. The inquiry is well aware that under the 2011 government pandemic strategy, we stockpiled large numbers of therapeutics, a large number of a therapeutic, I think Tamiflu, to cater for the possibility that the next pandemic would be a flu one. Of course, it was a gamble that failed to come off. There is obviously a very high level of risk in terms of stockpiling for disease X or the pandemic. You just don't know which one it's going to be. But is there nevertheless a huge importance in stockpiling something to cover a range of pathogenic outbreaks, just to be able to try to reduce at the outer edges the worst impacts of it?
Yes, and I think we are reasonably confident what the next pandemic will be. It will be influenza. It was for every decade except the last one. And I think we're all rightly concerned today about the potential for a devastating, or at least a bad, flu pandemic. And were avian influenza to become readily transmissible, we'd be in very, very serious trouble. So whereas I don't think we need to stockpile drugs for Ebola or Marburg or Lassa fever or exotic viruses, the one that is most likely to come is influenza, and we do know that coronaviruses may come as well. So those are two virus groups where it would be wise, I think, to have stockpile. Indeed, the country does have a stockpile for, I understand, for influenza, and that's a wise decision. What we don't have is the information about what to stockpile, for the reasons that we've just discussed at length.
But some therapeutics will have some effect, or are likely to have some effect, by way of a repurposed use. And therefore, it's essential to have something there to at least help ease the passage of the response to the next pathogenic outbreak.
Yes, for those two specific viruses, influenza viruses and coronaviruses, yes, we have drugs that should work.
Thank you very much.
Thank you, Mr. Keefe. Mr. Wagner. Mr. Wagner's over there.
Good morning, Professor White. My name is Adam Wagner, and I act on behalf of clinically vulnerable families who represent the clinically vulnerable, the clinically extremely vulnerable, and the immunosuppressed. I've just got one area to ask you about, and it relates to the different kind of drugs that were trialled during the pandemic and the resources that were allocated respectively to the different trials. So at paragraph 5.26 to 5.29 in your report, you discuss the large-scale trials, for example, Principle, that were established to evaluate the efficacy of repurposed drugs in the treatment of COVID-19. And you say that Principle eventually published results for 6 candidate drugs, but 5 of these showed no benefits in terms of reducing the risk of hospitalization and death. And then you say at 5.39, you refer to the efficacy of specific antivirals, so specifically designed antivirals, molnupiravir and Paxlovid, that were ultimately procured for the treatment of early COVID-19 disease. So my 2 questions are these. First of all, in light of those results, do you think that an appropriate balance was struck in terms of resources and time that were being allocated to the evaluation of repurposed drugs in comparison to the resources and time allocated to the development of direct acting antivirals for the treatment of COVID-19?
In brief, overall, yes. Because you have to think at the beginning we had nothing and then we had something. So initially, when we have nothing, we look for the most promising of potential repurposed drugs with a low expectation that they will be successful. But then, as the specific, if you like, bespoke antivirals develop, these have a high prospect of being successful. And so resources then shift towards the evaluation of those. But overall, my answer to your question is yes. I think the balance was in my opinion, reasonable.
And having had that experience of the COVID-19 pandemic and the success and failure of various developed drugs, do you think there are any lessons to be learned for the future in terms of that respective attention and resources? In other words, do you think that is precisely the model that will always be seen? At the beginning, you focus on one thing, you move on to another, or are there other lessons we can learn about different models of resource allocation to try and get better, more, and faster drugs available?
Well, I think we've already discussed that to some extent. The idea that you have a reasonable we have a reasonable prediction about what's coming, what might come, and therefore the probabilities of these things happening. And then you have things in-house that you think should work against those particular organisms. Now, you might need to modify those. And so initially you would repurpose or use the things that you already have, but then you would try and improve them. This is what the pharmaceutical industry does all the time. It takes a drug that works reasonably well and tries to make it better. So that process, I think, does work reasonably well.
Okay. Thank you.
Thank you, Mr. Wagner. And Mr. Thomas, who's just there, you'll see him when he stands up.
Good morning, Professor White. My name is Leslie Thomas and I'm representing FEMHO, the Federation of Ethnic Minority Healthcare Organisations. Professor White, in your report at paragraph 8.3, you state that as part of your recommendations, quote, an open forum for exchange of information in the relevant scientific community should be created with clear and capable leadership. Question. From the perspective of securing proportional representation, would you see benefit in this sort of open forum, including medical experts or healthcare leads from ethnic minority communities who can speak directly to ethnic minority interests?
Yes and no. So it depends entirely on the experience, capability, qualifications of those individuals. I really think it's important to have ethnic minority representation in some form, but it might be better to have, to have that in terms of community representation. For example, often the problem isn't so much of biology, but it's of access or behavior, and these people understand that better. So whether it's critical to have medical experts represented, I'm not so sure, but I do think representation is important in the way that is most effective, which perhaps you would guide.
Thank you. Can I move on to my second question? You talk positively in your report about the success of the recovery trial. For example, at paragraph 5.69 of your report, you talk about the proportional representation by ethnicity that trial was able to recruit. Specifically, you say, with regard to the UK's COVID-19 therapeutic trial, although not perfectly matched to the population, the representation was generally better than many other trials. Whilst the recovery trial did not report a detailed breakdown of ethnicities, the proportion of its participants who were non-white was similar to the UK population in the 2021 census, reflecting broad recruitment across many different hospitals. My question is this: how might lessons from the recovery trial be applied globally to standardise proportional representation in rapid clinical trials?
It's a very good question, and I think the UK is better placed perhaps than most to respond to that appropriately. So by simply spreading the net very wide, the recovery trial got a reasonable representation of the community at large. I think in other countries it is more difficult. It is an important point, but I'd just like to reiterate a point I made earlier. What really matters is speed, because everyone will benefit if we get an answer more quickly. And if there are underrepresented groups in the trial, then after getting the result, then you can then address that. Whereas slowing a trial down in order to engineer representativeness may be to everyone's disadvantage. So I think it's a very important point. I think the NHS was particularly well suited to engineering this, but I do think that speed is of paramount importance in an epidemic or a pandemic.
Thank you, Professor White. Thank you, my lady.
Thank you, Mr. Thomas. That completes the questions we have for you, Professor. Thank you so much. Have you flown in from Thailand especially for us?
Yes.
Well, thank you very much for going to that amount of effort. As you'll appreciate, there's an awful lot in your report we haven't been through today, but I will make sure that I am completely on top with the assistance of my team of all the matters there. And your report has helped inform our investigation and our questioning of other witnesses. So thank you very much indeed for your help.
Thank you, my lady.
My lady, may I just make plain, before I get a phone call from His Majesty's Treasury, that Professor White flew into Europe in part to attend this inquiry, but also for other professional purposes.
Thank you very much. I shall return at 11:20. Williams.
My lady, the next witness is Helen Knight. Please, could she be sworn?
I swear by Almighty God
I swear by Almighty God
that the evidence I shall give
that the evidence I shall give
shall be the truth,
shall be the truth,
the whole truth,
the whole truth,
and nothing but the truth.
and nothing but the truth.
Thank you for coming today and assisting the inquiry. You've provided a witness statement dated the 18th of November 2024 on behalf of the National Institute for Health and Care Excellence, also known as NICE. And the INQ for that is 000474611. Are the contents of that statement true to the best of your knowledge and belief?
Yes, they are.
Thank you. Just first touching briefly on your professional background. You are currently the Director of Medicines Evaluation at the Centre for Health Technology Evaluation at NICE.
Yes, correct.
And during the pandemic, you were Programme Director for Health Technologies at the start.
Yes,
And then Deputy Director and then Acting Director.
correct. Yeah.
In terms of the centre, is its role to undertake health technology evaluations?
It is, yeah.
And could you please explain, just in brief terms, what a health technology evaluation or appraisal actually is?
So at NICE, we conduct independent and evidence-based guidance to the NHS on treatment of new medicines and health technologies. And we make that assessment on a review of the clinical effectiveness and the cost effectiveness to provide guidance to the NHS.
Thank you. And I think it's right to say there are different forms of these evaluations, but during the relevant period for this module, the majority of the evaluations for non-COVID treatments followed the single technology appraisal process. Is that right?
Yes, that's right. So for medicines, yes, the majority of the work was single technology appraisals.
And does a standard one of these appraisals usually take about 44 weeks?
So that's an average. It varies sometimes between 39 and 60 weeks, but yeah, on average, 45 to 50 weeks.
So early on in the pandemic, was it recognised that there was a need to adapt NICE's usual ways of working to provide access to treatments for COVID patients as quickly as possible?
Yes, that's right. Yeah.
And ultimately, that's what led to the establishment of Rapid C19.
Yes.
I should say that stands for Research to Access Pathway for Investigational Drugs for COVID. Okay. Mrs. Knight, the focus of your evidence today is going to be on Rapid C19's consideration of the prophylactic drug Evusheld. But before that, I'd like to ask you just a few questions about the work of Rapid C19 more broadly. So, Rapid C19 was established in April 2020.
Yes.
And was it stood down at the end of March 2023?
Yes, that's right.
It was a multi-agency initiative involving, in particular, NHSE, MHRA, NIHR and NICE. And is it right to say its focus was on access to therapeutics within England because it's a devolved activity?
Yes, that's right.
But representatives from the devolved nations participated in the initiative and attended meetings?
Yeah.
Rapid C19's role was to identify and monitor the development of potential therapeutics and then assess their clinical effectiveness in order to facilitate rapid access. Is that correct?
That's correct, yeah.
Was this primarily through clinical trial evidence?
Yes, that's right.
And did Rapid C19 initiate, approve, conduct any trials itself?
No, we weren't responsible for kind of research initiatives or commissioning any research studies.
And is it right to say that unlike the usual technology appraisal process, the Rapid C19 process didn't consider cost effectiveness?
Correct, no consideration of cost effectiveness.
Could you just explain why that approach was taken?
Yeah, I mean, the initiative was set up as part of the temporary emergency circumstances of the time, and the focus was very much around identifying medicines for COVID that had a signal that they would be clinically effective and therefore important to roll out rapidly in the NHS once we were confident that they would work.
Thank you. You've set out in your statement how Rapid C19 went about its work. I'm not going to go through this in detail, but if I could just summarise in very broad terms. First, potential therapeutics were identified through horizon scanning of clinical trials and then ranked against various criteria to kind of prioritise them for consideration.
Yeah, that's right.
And then topic briefings were produced and kept up to date with emerging evidence.
Yes.
And was there an oversight group that then met to review these topic briefings and consider the therapeutics?
Yes, we did, yeah.
Were the senior members of that group the same representatives from the same organisations we've touched upon, so NHSE, NICE, MHRA, NIHR?
Yeah, that's correct.
It's a long list. And were you one of the representatives for NICE?
I was, yeah.
So is it right to say that the oversight group would meet and then decide, A, whether to recommend access, B, whether to continue monitoring the drug, or C, whether to deprioritise?
Yeah, so in particular on the recommendation, it was where we felt that we had seen or we could be confident that the medicine looked to work, and therefore we would provide that signal to the Chief Medical Officer for England and the Department of Health to be considered for rollout into the NHS.
And you do that by producing a report, a written report with an opinion for the Chief Medical Officer and DHSC?
That's right.
But ultimately, it was for DHSC to decide whether to actually go ahead and provide access to the drug.
Correct.
Just turning now to the therapeutics that were recommended for rapid access, I think, is it right to say that at the end of October 2022, the initiative had resulted in over 200,000 patients receiving treatments?
Yes.
And did Rapid C19 contribute to access for both repurposed and also new drugs?
Yes, it did.
I think there are 10 set out on your statement, so I'm not going to list them all, but did they include remdesivir, dexamethasone, sotrovimab, molnupiravir, and Paxlovid?
Yes.
And in your statement, you helpfully set out some tables which show the timescales for access to these drugs. We don't need to bring them up now, but is it right to say that for repurposed drugs, the majority of them were made available within a few days of the trial results, Rapid C19 becoming aware of the trial results?
Yeah, that's right.
And in fact, for dexamethasone, it was on the same day as the trial results?
It was the day that the trial results were published, so we were able to kind of patients in the NHS were able to access it on the same day.
And in terms of new drugs, which of course needed marketing authorisation, is it right to say that generally access was made to those within 2 to 6 weeks from authorisation?
Yes, that's right.
So I think it's fair to say that both repurposed and new drugs were made available through the initiative much quicker than would have been the case through NICE's usual processes.
Yes.
Mrs. Knight, in your view, what were the principal factors that contributed to Rapid C19 being able to achieve this?
I think it was key to have the right representation within the group. So as you identified, 4 key organisations that all are experienced in reviewing clinical effectiveness data, clinical trial data, but brought slightly different perspectives from the organisation. When we met regularly every week, we were able to have an open discussion, transparent discussions to make sure that we could make the best quality review of the data and support rapid access where it was where a medicine was shown to be effective.
And I think one of the other things you say in your statement is that the horizon scanning of the trials worked particularly well. Is that right?
Yeah, so it was a comprehensive undertaking from the team to actually monitor all of the ongoing trials for COVID medicines, and that wasn't just UK trials, it was worldwide. So that was a significant level of activity that was done on a daily basis.
Thank you. I'd like to turn now to Evusheld, because that was one of the drugs that was not made available through the initiative.
Yeah.
So the inquiry's heard a considerable amount of evidence about this prophylactic drug, and in particular the vaccine Taskforce early work and the ministerial decision not to proceed with it at that stage. Was Rapid C19 involved at that moment?
We weren't involved in those early stage discussions. So we first became aware of Evusheld from February 2021.
And that's when you first started to consider it?
Yeah, that's right.
And how did that come about in February 2021?
So my recollection was that this came via the ATTF at the Department of Health. So we were... it was signalled that there was a conversation with the company AstraZeneca, and that's when we had already identified it, but we brought that discussion within Rapid C19. We brought that forward and had the first discussion in February.
And its focus on Evusheld was as a pre-exposure prophylactic?
So it was being trialled for several different uses, but the key focus at Rapid C19, or the majority of the discussion, was around the pre-exposure prophylaxis.
And of course, as the inquiry has heard, the significance of this is that could benefit those who might not be protected by a vaccine.
Yeah.
So just looking at the period between February 2021 and November 2021, Rapid C19 essentially at this stage were just noting the ongoing trials and monitoring them for any key results.
Yeah, that's right.
And then it was in December 2021 that there was trial evidence which first suggested it might be effective.
Yeah.
So if we could just turn to the report in December 2021 then, and that's INQ 000479901. Thank you. And we're on the right page already, page 12. We can see there that's the report to the CMO dated the 23rd of December 2021. We've got the various names for Evusheld. You can see the senior Rapid C19 members, and it includes, there's expert advice from the prophylaxis oversight group. We'll come back to that group in a moment. We can see that it's Rapid C19 is reviewing the trial results from Provent. And if we could go straight to the recommendation at page 16, please. So we can see there, the second bullet point, it says Evusheld is the first medicine to show robust benefits in pre-exposure prophylaxis. And then moving down, please, there's some unanswered questions about the results, and we can see in the middle of that it says the prophylaxis oversight group noted that there's a risk associated with introducing a partially or minimally effective therapy. Do not currently recommend routine use of this treatment until more data on efficacy against Omicron are available. And then at the bottom, thank you, overall, there's reference there to strong signal of efficacy that warrants action to prepare for access subject to first marketing authorization and second, confirmation of continued activity against Omicron.
Yeah.
That can come down for now, thank you. So at this stage, the main concern was whether Evusheld was effective against the Omicron variant.
Yes, that's right.
And was this an issue that not only affected Evusheld, but actually other types of neutralising antibodies?
Yeah, that's right.
I think, in fact, you say in your statement that changes in effectiveness due to the variants was one of the key challenges for your work.
Yeah, it was.
Could you just explain what the implication was for this in respect of clinical trial evidence?
Yeah, so the key challenge really was, although that the trial data would show that there are there's clinical effectiveness in terms of protection, what we couldn't be sure of is if that same level of protection would be seen in clinical practice when different variants were circulating in clinical practice versus the variants that were circulating during the trial period.
So essentially, you couldn't be sure it was effective at the time you were actually looking at considering access based on what might have been outdated trial results.
Yes, that's right.
And was there a difficulty with getting updated clinical trial evidence?
Yeah, I think we recognise that kind of repeating those clinical trials would be a challenge, particularly with the way that the virus was changing. That we would be presented with that problem moving forward.
I think one of the other things you say is that there was little appetite from commercial sponsors to undertake further trials.
Yes, that's right.
So then were in vitro, and when I say in vitro, I mean lab-based studies, were they the quickest way of trying to assess any ongoing activity against the variants?
Yeah, so obviously, and you've referred to, we invited the prophylaxis oversight group experts to come because we recognised within Rapid C19, which was set up to assess clinical effectiveness data, we didn't have the necessary expertise. And with the neutralising monoclonal antibodies, it became clear that actually in vitro data was going to be an important consideration for these products.
And just in relation to the lab-based studies, you obviously said that Rapid C19 didn't have the necessary expertise. But is it right that another issue with these studies is that they were quite difficult to interpret and to extrapolate from the lab test to clinical effectiveness?
Yeah, what we heard at the time is that there were no real standard methods for assessing the quality of the in vitro studies and the results of those in vitro studies. And we didn't have any scientific consensus on how in vitro results could actually be interpreted into clinical effectiveness outcomes.
Thank you. Just moving on then to May 2022. I think there was an oversight group meeting on the 18th of May, and that resulted in another report to the CMO
Yes.
on the 30th of May 2022. Could we have back up that same document, please? And this time turning to page 8, thank you. So is it right to say that at this point, conditional marketing authorization has been granted, and you now have the in vitro data from UXA and also from Oxford University, but I think the focus was on the UXA data.
Yeah, that's right.
Turning then to page 10, which is where the recommendation set out, thank you. At the second bullet point, it says, Rapid C19 does not consider that the available non-clinical data supports clinical effectiveness. It refers to difficulties in interpreting the data, and I think that's the point we were just discussing.
Yeah.
And then if we go down to the last bullet point for now on this page, essentially, because of the difficulties, is it right to say that there was no certainty that Evusheld would work? And there was a particular concern about the population that might get Evusheld in terms of their vulnerabilities.
Yeah,
And you can see just at the bottom, it says the risks of proceeding to access are considered to outweigh the risks of not providing the treatment.
yeah.
Could you just explain that a little more?
Yeah, of course. I mean, we were very aware that we were discussing, a prophylactic medicine for a patient, patients who had a high unmet need, but were also very clinically vulnerable and shielding at the time. And so we felt, these were people that weren't infected with the virus, and therefore, we wanted to make sure that we had a high confidence that the treatment would protect this clinically vulnerable group, because, we wanted to be confident that that protection would continue, and we didn't see the evidence to say that it would.
Thank you. And just staying on this page, moving up slightly, it's recognised it's not feasible to obtain clinical effectiveness data. There's a need to understand how non-clinical trial data, so that's the lab tests, could be used to support decision-making on clinical effectiveness in the face of the variants, essentially. And then it also says it's going to be an ongoing issue for these types of drugs. And then, thank you, if we could turn over to the next page. Thank you. So at the top, we've got the ultimate recommendation, which is not to proceed, but under the proposed actions and the final bullet point, Rapid C19 will contribute as needed to system-wide work to consider what evidence is required to be confident, and the Antivirals and Therapeutics Taskforce will be taking this work forward. Thank you. That can come down now, thank you. What type of work is being referred to here?
So what we were trying to understand was, exactly what we said about creating, standardised methods for actually understanding the quality of in vitro studies and therefore the results that you can take from them. And more importantly, how can you then translate in vitro results to the clinical effectiveness of a medicine? So they were the particular areas that we were wanting to be explored.
Okay. I think you will have seen in his report, or probably heard this morning as well, Professor White raises the issue of pharmacometrics. And I think he explained this morning that's the combination, forgive the technical detail, of both pharmacokinetics and pharmacodynamics. His view is that this type of data may have answered any unresolved questions about Evusheld's efficacy. Did Rapid C19 consider getting this type of data?
So during the discussions at Rapid C19, and again, acknowledging we round the table weren't experts in that field, we had the prophylaxis oversight group come to help us. The term pharmacometric study was never raised or discussed. We talked about the neutralising effect that you can get from in vitro studies, and we referenced more PK/PD analysis, but pharmacometric studies was not something that was ever raised or discussed. I don't know that would actually answer the key question that we were looking at, which was how can you translate in vitro data into clinical outcomes. I'm not sure that pharmacometric studies would actually answer that question.
Thank you. In terms of the work that was discussed, are you aware whether the Antivirals and Therapeutics Taskforce took this forward?
So NICE was invited to a roundtable in September of 2022 to start some discussions. There was a brief follow-up meeting, but I'm not aware of anything further that happened.
Just pausing there for a moment, because I think we're going to come on to the final report in the chapter, which is in August 2022. But between May and August, were you asked to contribute to any further work on this?
So we were aware of the potential considerations of the kind of real-world evidence studies that were being conducted in other countries. And we were also considering and having discussions about whether or not there may be a potential for Evusheld to be enrolled in one of the platform studies, in particular Protect V. But other than that, I'm not aware of anything else that happened.
Just in terms of Rapid C19's role, I think one of the other things that Professor White says in his report is that the various committees were generally passive in that they did not direct or initiate research. Insofar as that might relate to Rapid C19, do you think that's a fair assessment?
I mean, I wouldn't necessarily say Rapid C19 was passive. I mean, it was a significant undertaking working proactively to identify medicines for Covid-19. I think what we tried to do was signal when there was clinical effectiveness evidence to roll a medicine out, and where there were gaps in the evidence, we certainly signalled that for consideration within and I think you can see that in the CMO reports.
Well, on the CMO reports, we're going to come to the last one now, in August 2022. And at this point, is it right that the ATTF have asked you to look at real-world evidence from countries which had deployed Evusheld?
Yes.
Okay. The same document again, please. Page 1, thank you. And at this point, we can see the prophylaxis oversight group is still providing experts' advice. Again, just turning to page 6, please, for the recommendation. At the bottom there, thank you. Essentially, the evidence is still insufficient at this stage to progress. Yeah. And then looking at the proposed actions, please, on the next page. So here, Rapid C19 is recommending further research and is recommending pharmacokinetic analysis. Do you know whether this work at this point was taken forward?
So this, because this report was in the August, I think that was then what led to the roundtable that was taken forward in the September by the ATTF.
Could you just help the inquiry understand? Obviously, we can see here that you are recommending further research and also specifically pharmacokinetic analysis, but didn't recommend that back in May. Can you explain why?
So I think, from the notes, I wasn't actually at that May meeting myself, but, I think, had it been a key area for consideration, we would have flagged that for the ATTF to consider. I mean, I think with pharmacokinetic and pharmacodynamic data, it requires, the drug to be able to understand that. And of course, the Rapid C19 members were not in control of accessing the treatment, nor were the other research organisations.
Is it right by this point that NICE then receive a referral from DHSC to conduct a full technology appraisal of Evusheld, and ultimately it's not recommended through that process either?
Yeah, that's right. But if I can just also add, during kind of that summer period, I mentioned that we were potentially exploring whether Evusheld could be used within the platform study Protect V, and really that, we felt that would have provided an opportunity to somehow link in vitro data to actual clinical outcomes. But as I said, unfortunately, that was never taken forward.
Are you aware why it wasn't taken forward?
No, I mean, again, it would require the company making the treatment available for us to roll out that in a clinical trial.
So just looking overall at the picture between February 2021 and August 2022, to the extent you've not already answered this, do you think Rapid C19 could have done more to consider the effectiveness of Evusheld?
I think we responded as quickly as possible to all the new available data. you can see from all of the reports, we considered it extensively. And as I said, because of the particular patient population who we were talking to, we detailed that we required a high level of confidence to be able to signal that, this would be considered for use in the NHS. And ultimately, we didn't have that confidence from the data.
I think this is probably self-evident from your last answer, but did you have in mind when making these decisions just how important a possible prophylactic treatment for these type of groups might be?
Absolutely.
Just in terms of the overall system more broadly, not just rapid C19, if we could just get up paragraph 136, I think it's at page 55 of your statement. Thank you. We can see there that you discuss the European Medicine Agency's emergency taskforce statements on antibodies and Evusheld and the FDA's suspension of Evusheld in the US. You then go on to say that NICE considers that with the benefit of hindsight, the UK public health and regulatory system could have looked more intensively at whether or not Evusheld was effective against SARS-CoV-2 variants. Could you just help us understand which parts of the system you're talking about and how you think they could have looked at this issue more intensively?
Yeah, I think here this is, it became clear that these neutralising monoclonal antibodies were very much impacted by the changing virus. And, I wonder whether more could have been done to help try and create that link between in vitro data and clinical outcomes. As I said previously, when Rapid C19 were considering this information, there were no standardised methods to assess the quality of that those in vitro studies. So potentially that was something that would have helped us understand the confidence of that data. But equally, the virus was changing so quickly. It would be difficult to keep up with that, and I also don't know what resources the UK HSA and the MHRA would have to be able to track and monitor that.
But for the future, do you think that's something that those organisations should be looking at?
Yeah, I think it should be explored.
Thank you. And just on lessons for next time, I think one of the things you refer to in your statement is an in vitro expert advisory group that's been commissioned by NICE. Could you tell the inquiry a little bit more about that, please?
So at NICE, we stood up an in vitro advisory group to help inform the technology appraisals that we were to conduct as part of the routine consideration of these COVID treatments. And in the absence of data, we pulled together a representative group of experts, and that discussion and framework helped us understand the neutralising effect of the monoclonal antibodies and how that could be considered within the appraisal and translated into clinical effectiveness.
So has it actually made a practical difference to NICE's work since the pandemic?
Yeah, I mean, like I said, in the absence of anything, because we knew we were going to have to undertake the appraisals, we felt we needed to, to provide that information to our independent committees. Was it perfect? I don't know. Is there more that could be done? Potentially. But, it was very clear when we looked at the medicines at that time that the neutralising effect in some was either reduced or absent, and that really helped to guide the committee's decision making.
So do you think that could be helpful in the face of a future pandemic?
Yes.
Are there any other improvements that you think could be made around research on this issue?
I think we've obviously touched on the key aspects already. I think it would be helpful to consider how we might be able to have more diverse people included in clinical trials. We didn't really see very much data, particularly on those clinically vulnerable groups. There may be a challenge, there may be ethical issues with that, but more understanding on how differently or not a medicine might work in those groups would be helpful, and in particular exploring medicines in groups of people who we think won't respond to the vaccine or can't have a vaccine.
In your statement, we don't need to get it up, but you say, just in terms of development for therapeutics, that the system could do more. Is that essentially what you've just touched upon in terms of the types of trials and the types of participants in those trials?
Yeah, and also, we obviously, particularly for the monoclonal antibodies, we know that the effectiveness can be impacted with different variants. So maybe in the research and development, actually trying to identify, the particular spike proteins that they will work on to try and get a broader, to maintain a broader effectiveness even when the virus changes. It's not my area of expertise, but I wonder whether it is something that could be taken forward.
Thank you. My lady, those are my questions.
Thank you very much. Ms Douglas has some questions for you. She's over there.
Thank you, my lady. Good morning. I act on behalf of clinically vulnerable families who represent the clinically vulnerable, the clinically extremely vulnerable and the immunosuppressed. You have explained in your statement and also touched upon earlier this morning that the rapid C19 process did not include consideration of cost effectiveness, and that was appropriate in responding to an unprecedented public health emergency for the focus to be on the clinical effectiveness and not the cost effectiveness. You've also explained that approach was a deviation from the standard routes of access to therapeutics available in the NHS. But we've seen, of course, that the ultimate decisions in government regarding Evusheld did cite cost effectiveness. As an example, Sir Sajid Javid has said that the advice he was given in June 2022 was that Evusheld was too expensive for the advantages that it would give. And so my question is this: if NICE's primary purpose ordinarily in routine times is to conduct a cost effectiveness analysis to ensure value for the taxpayer and the financial sustainability of the NHS, how did you ensure that those cost considerations did not end up influencing your discussions, advice and recommendations about COVID-19 therapeutics, and particularly Evusheld?
Yeah, obviously I attended those meetings and we never considered the cost, particularly with Evusheld. We were very much focused on understanding the clinical trial data, which obviously was positive, but how that would translate into clinical effectiveness in the NHS. We didn't consider cost for Evusheld in the same way that we didn't for all of the other medicines that we were considering. Some obviously which did show effectiveness and then were rolled out in the NHS, and others that didn't show effectiveness and therefore weren't. So at no point were we thinking about cost or cost effectiveness in our kind of discussion on the evidence and our advice up to the CMO.
Thank you. And so you would have no concerns that those types of considerations influenced any of the ultimate advice?
In Rapid C19, there was no consideration at all. I can't obviously comment on conversations that may have happened elsewhere, but I can confidently say that, it was not a discussion point within the Rapid C19.
Thank you. Thank you, my lady.
Thank you, Ms Douglas. That concludes the questions we have for you, Mrs Knight. Thank you very much indeed for your help to the inquiry and coming along today. And thank you also for the work that you and your team did. I think the horrid modern word is pivot, isn't it? But for getting the process done more smoothly and more efficiently, because we're in a national emergency. So thank you very much indeed.
Thank you.
Mr Mansell.
My lady, the next witness is Dr. Clive Dix. If he could be sworn, please.
I do solemnly,
I do solemnly,
sincerely and truly
declare and affirm
that the evidence I shall give
that the evidence that I shall give
shall be the truth,
shall be the truth,
the whole truth,
the whole truth,
and nothing but the truth.
and nothing but the truth. Thank you.
Sorry if we kept you waiting, Dr Dix, but you are, I think, our last witness.
That's right, my lady, the last witness of this module. And Dr. Dix, could you give your full name to the inquiry, please?
Clive Dix.
Thank you very much for coming today to assist the inquiry. You have provided a witness statement for Module 4, that's INQ 474423. It's signed by you, and is it true?
Yes.
Thank you. You served as Deputy Chair of the Vaccine Taskforce, or VTF, under Dame Kate Bingham from June to December 2020.
Yes.
We've heard from Dame Kate. I don't wish to embarrass you, Dr. Dix, but she described you in one word, phenomenal. But there we go, perhaps I have embarrassed you.
Yes,
and then you served as Chair of the VTF from December 2020 to April 2021.
That's correct.
Briefly, just going back through your education and career, you graduated from Leeds with a first-class degree in pharmacology.
That's right.
Following which you obtained a doctorate.
True.
And after several postdoctoral fellowships at the Royal Free Hospital in London, you moved into the pharmaceutical industry.
That's correct.
Since 2001, you have held several Chief Executive Officer, Chairman and Non-Executive Director posts at a range of pharmaceutical companies, specialising in vaccine research and development, some of which you co-founded.
Yes, that's correct.
And you explain in your statement that you have significant experience and knowledge of the pharmaceutical industry and the finance community supporting the sector.
That's right.
And it was no doubt that experience that you drew on in your work with the VTF. And the first topic I'd like to ask you about, please, is your role as Deputy Chair of the VTF, particularly in building the UK's vaccine portfolio. Is this right, that on joining the VTF, your role was to build and head a team which would identify the most promising vaccines currently in development?
That's right.
How prepared, in your assessment, was the UK to undertake such a task?
It didn't have any infrastructure or thinking at the time at all.
What about the links between the government and industry? What state were they in when you took that role on?
They were sort of transactional, to do with buying things, and very, I would say, adversarial. There weren't fabulous relationships, partly because of things like drug pricing, which tended to be adversarial events anyway.
The aim was to have a diverse portfolio of vaccines, including vaccines based on different technologies.
Yeah.
You were working on a list of around 200 vaccine candidates.
That's
initially. Right. How did you go about the task of narrowing down that list?
It was a very rigorous process, the sort of process you use when you're looking at science generally in pharmaceutical companies deciding what to do. So we had access to a thing called Airfinity, which was a database that was a brilliant database that was continually being updated on what was going on in the world of vaccines. And then we set a set of criteria of which we looked at each of the vaccines in turn and decided whether they met those criteria, and then moved on. So it was a set of filters, if you like, and that honed it down quite quickly. One of the key filters was, could this vaccine ever be in a human being to be tested within that year, so by the end of December 2020. And that took a lot of them out, basically. So it wasn't a difficult process to get to a smaller number. But then what we had to do was meet with the businesses, the companies that were making these vaccines. And so we set up NDAs with all of them, and we contacted them, built relationships with them, and asked them to show us all of their data so that we could analyse it together as a team and look at it from all the different aspects and decide whether it was a going concern, really.
You explained that the list of 200 was reduced through applying this criterion to around 15 before you approached the various companies.
Yeah.
And at that stage, you were carrying out what you describe as deep due diligence. So what did that involve?
Well, that involved asking the company to let us into all of their documents and see every piece of information that they'd already gained on these molecules, and also what their plans were. So we judged whether their plan was going to work, and whether we did a lot of detailed analysis of their manufacturing capabilities, and we saw where the weaknesses were and the strengths. And in fact, in all of them, there was a chance then to say, well, we can help you with some of this because you haven't quite got it right. And then we brought in experts that would help with the clinical trials sometimes help with manufacturing, and linking them to the MHRA so they could get advice on what was required for the full clinical study. So it was just detailed analysis of the science behind these vaccines.
Would that result in a report that your team put together on whether a particular company or vaccine candidate was a promising one, which you would then present to the Vaccine Taskforce steering group for a decision ultimately to be made
basically
on whether it would be backed?
every single vaccine had a diligence report with a recommendation in it that went into the steering committee, and eventually, if it became the one we took forward, it went as far as the ministerial panel.
You would then look at what could be done to incentivise development of a particular vaccine. You've touched upon this, but clinical trial support, providing and investing in manufacturing facilities and fill and finish capabilities for that particular candidate.
Right from the outset, Kate and I sat down and talked about how can we be the first? What's it going to take to make the vaccine industry come here rather than go to America, for instance? And so we basically were salesmen on this is the best place to do it, so we can show you what we can help you with. And we did, and we put things in place, and clearly that worked.
Being the best client.
Yeah.
That 200 list of promising or vaccine candidates initially went down to 15, and then the 7 in the vaccine, the VTF's portfolio. There came a point in December 2020 when you took over as chair of the VTF.
That's right.
Now, I don't want to go into the details, but suffice it to say you were not happy with how the role of chair was framed post Dame Kate's position. And in particular, you were concerned about the lack of having a single point of accountability to a minister, which Dame Kate had and wasn't going to be the case for you.
Absolutely. I got a letter of appointment which didn't have that, and I then rewrote my letter of appointment with a bit of help from Kate, as it happened, and asked for this to be considered, and it was considered and rejected. So it became very difficult because we knew that the success of the VTF at that point was basically down to that single point of accountability and the ability to ensure that the steering group really did scrutinise all the recommendations and have all the right expert input before it went to ministers.
Why was that so important, that single point of accountability to a minister?
Because you wanted to ensure that all of the input that came from the Vaccine Taskforce, from all of the experts as well as the government officials, was sound, and it was brought together as one, and that there wasn't any room for, manoeuvre into the wrong areas. So it was important to ensure it was crisp and clean and moved fast.
You stepped down in April 2021 as chair, and soon after that, Sir Richard Sykes became the chair of the VTF?
That's correct.
The inquiry has heard a great deal of very positive evidence about the work of the VTF and what it achieved. And I'd be interested in your views, please, on whether the key features are captured in a document authored by Lord Valence. Can we have on the screen, please, INQ 101626? This is a document which was authored by Lord Valence, titled Key Features of the Vaccines Taskforce, dated the 10th of February 2021. And we can see here that he's set out the 7 factors which he's highlighted as being the key features which led to the success of the VTF. Content experts were brought in rapidly. Number 2, an at-risk investment mindset was taken. Number 3, procurement was part of the process.
Yep.
Number 4, there was a very clear and measurable outcome objective. Number 5, a single point of accountability for the whole activity, and that person was empowered and answerable to the PM. So the point perhaps you were making a moment ago.
Yep.
Number 6, private sector engagement was key. And number 7, long-term legacy was built into the thinking from the beginning. Do you agree with the analysis set out in this document? Does this capture what made the VTF so successful?
Absolutely, and the long-term legacy was at the forefront of the minds of everybody as we were doing the deals with the companies, so that can we attract them to be here as part of the future, and therefore part of preparedness for the future.
Thank you. That document can come down. And it really is that long-term legacy that I'd like to turn to now, Dr. Diggs, okay. Because in your statement you are highly critical of the UK's current preparedness for the next pandemic. You say the UK is in a weaker position now than it was prior to the COVID-19 pandemic. You say we have less resilience now and have failed to learn the lessons from the pandemic. And I just want to focus on some of the specific criticisms you make, with a focus on the lessons that can be learned, please.
Okay.
The first topic is Valneva, and the inquiry has heard that the contract with Valneva was terminated in September 2021, and this matter has been raised with a number of the Module 4 witnesses, including Professor Dame Jenny Harries, Sir Sajid Javid and Nadim Zahawi, and I'm just going to briefly encapsulate what's said about that, and then we'll get your view, please. But in essence, it said that the advice, which was from the VTF, although given after you had left, was that the contract should be cancelled because trial data suggested the Valneva vaccine would be ineffective as a booster dose. It was appropriate for the UK government to try and achieve value for money for the taxpayer by cancelling the contract in light of that trial data, and that the relationship with the company was considered as part of the decision, and the cancellation has not damaged relations with industry. What do you say?
Not that. In terms of relationships with industry, it was devastating, I would say. Devastating for the company, but also the ripple effect from that to other companies, that this was the way they were treated. That was the first thing. The second thing was that the data they used to make that decision was poor. It actually wasn't a full study. It was a study that was done in a rush to see if we could get booster data because we wanted to boost by the end of the year.
And just pausing there, this was the COMCOV boost study.
And they only left 2 months, 2.5 months between primary vaccination and boost. And anybody that knows about vaccines and vaccine development would know you would not see significant boosting at that time. So it was putting a vaccine through a test that was going to be negative. And given that the government had funded the study, the phase 3 study, which was about to report out, 6,000 people that the MHRA had put together the criteria that needed to be passed for it to be approved. And that study was going to read out 1 month after that decision was made. That study showed the vaccine was better than the AZ vaccine.
So the final
and it
data
was then,
from the study, did it contradict the COMCOV boost study?
Well, it doesn't contradict. It was just a very poor study. So you know when you do vaccine development that you do some small studies, and sometimes there's a bit of a red flag, or sometimes they look really good. But you don't suddenly stop because of that. You wait and see the real, proper, well-controlled, large studies and get the full readout. And when that readout came, it showed that the vaccine was as good as, and probably is going to be one of the better vaccines if it was still around even today. And what still perplexes me is the phase 1 and phase 2 studies which had been done and were available were not looked at for that decision. This small, what I would say was a flawed booster study, was the only study that was used to make that decision. And that was, I don't believe any experts were part of that analysis. I just can't believe there's an expert that would ever make a decision based on that data.
So who conducted the flawed analysis?
It wasn't the company. It was a professor, Saul Faust at Southampton, and it was done under a UK research group's funding. They did it across all the vaccines because they were looking for ones that would definitely be best for boosting. And unfortunately, some of the science says that once you've vaccinated people with an mRNA vaccine, the level of antibodies are so high, until they've fallen down, you shouldn't even try boosting because it's probably going to be very difficult to boost. So it was just a flawed study.
This advice had come from the VTF. You weren't the chair at the time.
But I don't think
it had come from the VTF. So in terms of who was looking at it, the VTF had looked at it.
Clearly.
Had something changed in the approach the VTF was taking?
Well, I heard some of the evidence that the experts around the table agreed. Well, unfortunately, I know all the experts because I worked with them. None of them were consulted.
These are members who were of the VTF.
Well, they were the diligence team, basically, the people that knew the vaccines inside out and were part of the people that you would consult when you made big decisions. And none of them were ever consulted on this decision. So I actually don't know who the experts were, and I didn't see any experts in the VTF once I left.
Moving on, next topic, another issue about which you are critical is the Vaccine Manufacturing and Innovation Centre, or VMIC, and the inquiry has heard that VMIC was sold in April 2022 and has subsequently been mothballed. You characterise that as having a serious negative impact on the UK's ability to develop and manufacture vaccines at speed in the face of a future pandemic. The inquiry has heard evidence about the new strategic partnership with Moderna's Innovation and Technology Centre, and the AstraZeneca investment in Speak Liverpool. Do those projects allay your concerns?
No. It is very good that we've got a manufacturer of an RNA vaccine because they will be part of the future without a doubt. On their own, they're not the answer, though. The AstraZeneca investment actually hasn't happened, and it seems to be faltering. But what needs to be made clear, I heard a lot of evidence about manufacturing, and I think some people talked about having sovereign manufacturing. The UK government does not have to invest in manufacturing and shouldn't invest in manufacturing, particularly for a pandemic. It's impossible for the government to be running state-owned or even state-invested manufacturing. It's not the future. VMIC was an example of something that went wrong. VMIC was a vaccine innovation centre. The idea was that when small companies that make vaccines or are developing vaccines want to move through to clinical trials and then move on to manufacturing, they don't normally have the wherewithal to know how to formulate those vaccines properly. And the Vaccine Manufacturing Innovation Centre was there to encourage that to happen and therefore bring people to the UK because it was going to be a centre of excellence. There was a decision which I didn't particularly agree with, I wasn't part of, but to actually turn it into a manufacturing centre, which is a very manufacturing is very cut and dry. It's hard, grungy work. It's not innovative. And they tried to mould the two into one into that centre, and it sort of went astray. And then the whole thing got sold. The innovation piece got lost, and that's the piece that the country should still have. That innovation centre was a fantastic idea, came out of the Office of Life Sciences strategic reviews, and we've lost it. But coming back to manufacturing, what we need is the ability to have really good relationships with the pharmaceutical industry, show them, like we did at the beginning of the VTF, show them what the UK can offer and encourage them to come here and do their research, development, clinical trials, and then manufacturing, and have manufacturing as part of their remit in the UK. And then you've got manufacturing, and it's run by them, it's paid for by them, and the government can just be there incentivising and understanding it, not trying to run it. And I think that's really important. It's a bit different to saying we need to invest in manufacturing. I don't think we do.
So the state doesn't invest in manufacturing. Instead, you make the UK an attractive place for companies to invest in and build manufacturing facilities here. That they run, and presumably you then have step-in rights in the event.
Step-in probably not necessary, because if you've got good relationships, you can say that, make sure you've got a line that's available, and you can go all the way to step-in rights if you want, but I don't think that's necessary.
So how do we do it? How do we make the UK attractive for that type of investment?
Well, we do the sort of things that we did with Valneva to start with. We helped them with the clinical trials. We helped extend their factory because they hadn't got enough cash at the time. And if that was still in place now, we would be selling vaccine to the rest of the world, which would have fitted our second objective. And we'd have had something as part of a legacy for the third objective. So it's having the vision and understanding of how you can do that and do it over and over again. And we have all the right elements. The vaccine registry needs extending. The MHRA probably need to think about having a more dedicated vaccine unit within there that understands regulation and knows how to deal with the vaccines all the time. And I think we need this vaccine agency.
And we will come to it, Dr. Dix, I promise. The recommendation you made in December 2020 for a national vaccines agency. But just before we move away from this issue of VMIC and investment in manufacturing, I've made reference to the Moderna Innovation and Technology Centre. Do you share a concern that others have about an over-reliance on mRNA technology when it comes to vaccines?
Absolutely. So there's rose-tinted glasses for mRNA because it was so good in the pandemic. It was a brilliant vaccine for an emergency. It's far from a perfect vaccine. At the moment, the cold chain is terrible and storage is difficult. The duration of action of these vaccines isn't great. 6 months, it seems to wane. The cellular immunity, which is a classic important part of an immune response for vaccines to give you long-term immunity, is weak. So they will get there, but they still need developing to become the perfect vaccine, but they're not there yet. And then when it comes to pandemic, resilience for pandemic will not come from just having RNA. I can paint a picture of a virus coming our way that mRNA will not work for. And that's not what we want. We don't know what virus is coming. But if it's a virus that we haven't done a lot of background research on, like we had with SARS and MERS, and we knew the best protein, the spike protein, was the antigenic protein. If we don't know that, an mRNA vaccine won't work. What you've got to do is take every single protein on that virus and develop every one to see which one works. That's a huge amount of work, and it takes a long time. I've always said that the reason we had Valneva, it's a live inactivated virus. For me, it was like crown green bowls. You always have your backstop. So if everything else goes wrong, you still put that one there behind, and you may still win. Valneva was that. If a virus comes along we don't know a lot about, all we've got to do is condition it to grow in cells, and we could start manufacturing it as a whole virus and then just inactivate it. It may not be the future sexy technology, but it works, and we've eradicated polio. We use a very similar technology for flu at the moment. It works, and it's trusted, and it's safe, and it's less reactogenic than all the other vaccines. So there's nothing wrong with it. It's just old-fashioned, and people don't like old-fashioned, but it works.
Next topic, please, is the monoclonal antibody cocktail Evusheld. We've heard a lot of evidence about Evusheld. We've heard some of it today from Helen Knight. The story is really in two chapters. The first is about the VTF's position and the OCMO advice, the CMO and DCMO advice in late 2020, early 2021. And the second chapter is on rapid C19. That's what Helen Knight was just giving evidence about. But focusing on the first chapter, the inquiry has heard from Professor Sir Chris Whitty and Professor Sir Jonathan Van Tam about Evusheld, and I just want to sketch out their rationale for the decision-making that they made, that they fed into, the advice they gave, so you can give your response. But they say in essence this, that the clinical picture on Evusheld was unclear. There was no clinical trial data, no information on side effects. Indeed, it was not authorised by the MHRA until the 17th of March 2022. It was not clear to whom it would be given, because even those with a degree of a compromised immune system can benefit from a vaccine. It would be wrong to compare it to purchasing vaccines at risk, because they're very different products. Unlike vaccines, you have to keep giving Evusheld over and over again, and it's less likely to be effective as the virus mutates. And finally, it was a lot more expensive on a per-dose basis than a vaccine, and had a short shelf life, which could lead to wastage. That is the rationale underpinning the advice that was given. What is your view?
I actually feel most of those are excuses, and the actual reason that it wasn't purchased was cost. And I'll take you through the thinking. So shelf life is an error. The shelf life of we did all the diligence on these vaccines, by the way, on these antibodies. The shelf life was, at the time when we put the recommendation in, already 6 months, but with a statement that all of their antibodies in this category in the past had shelf lives of over 18 months to 2 years, and that they were likely to have the same. The shelf life isn't even an issue, because you don't get a medicine of any type into your hands until it's approved. And once it's approved, you agree a delivery schedule to have it at the rate that you can use it. So that just isn't an argument. It's fatuous. In terms of giving it over and over again, this antibody protected people for 6 months. And we were talking about buying enough to give people 2 doses to get them through this early stage of the pandemic and free them from lockdown. So I don't understand over and over again. 2 doses, 6 months. And then there was some discussions about it being difficult to deliver to the patient, and they needed specialist clinics. It was intramuscular. It was an injection in the bum. These arguments sound great, but I honestly don't believe they're valid at all.
You were in post when this advice was being received.
I think I was leaving at the time, so it was right towards the end. Yeah.
I mean, do we have a sense of a wider view in terms of prophylactics from this document, please? INQ 66712. This is an email from Charlotte Taylor regarding astronaut data on prophylaxis. Charlotte Taylor, of course, was the acting director of the Antivirals Taskforce and Therapeutics Taskforce, and she's relaying here a conversation she's had with Lord Vallance about trial data on Evusheld as a prophylactic. And she says this: I've had a brief conversation with GCSA, including the astronaut data on prophylaxis. I said that there is limited enthusiasm for prophylactic use across the system. His reply: I think that is misguided. There is a clear place for them, and it just needs to be defined. And that can come down, please.
Yeah, I agree.
Does that accord with your experience?
Yeah, absolutely.
A lack of enthusiasm for prophylaxis?
The Vaccine Task Force had a very entrepreneurial way about going about things, and very much getting things done. And the prophylactic antibodies moved into the therapeutic space rather than stay within the vaccine task force, and therefore it started to be looked at in a very different way. And so, yes, there was less enthusiasm. Interestingly, though, Professor Jonathan Van Tam was part of the VTF and was very enthusiastic about us getting the antibodies to start with, and was a sponsor, if you like. So I don't know what changed, but in the end, the only thing I can see that's consistent is cost.
Antivirals. The VTF looked at whether to include antivirals within its remit. You had experience of antiviral drug discovery and development, but it's your view that including antivirals in the remit of the VTF would distract from the task in hand, and you note that antivirals need a very long development time and extensive long-term safety data to be approved. Now, in a context where everything is being thrown at vaccines in order to expedite normally long development timelines, why couldn't the same approach have been taken to antivirals?
I think it could, but it would have overwhelmed the VTF, and I don't think we'd have been successful. One of the things you learn when you run particularly small businesses, unless you focus, you die. You've got to focus, and you've got to keep that focus really honest. And antivirals is a completely different set of skills. We would have had to have a completely different set of people looking at them, and it would have made the VTF's role rather complex, I would say.
That's a case for not including them within the remit of the VTF.
Yeah.
Should antivirals have been looked at in a more intensive way by a VTF-like body tasked with that, or were they?
I think so. I think it could have been. They are still very difficult things to find, and it does take a long time to develop them. So unless there's something in development already that happens to flip over into COVID use, then it would take a long time. So it didn't look like it was in the same time frame as the vaccines.
Final topic, Dr. Dix, is recommendations, and we are there. We're at the National Vaccines Agency, which I know you're keen to talk about. And can we have on the screen, please, INQ 330659. This is a recommendations document from December 2020 that was co-authored by you and Dame Kate Bingham. Is that right?
Yes, and a lot of input from the Vaccine Task Force.
We can see at the bottom of that page that you recommend the creation of a national vaccines agency, and it states that it's clear that having the ability to draw together networks is crucial to pandemic preparedness. Just pausing there, that is perhaps a key lesson, is it, of the VTF, bringing together industry, academia and government? Yes?
Yes, absolutely.
You state the VTF recommends the creation of a new executive agency within BEIS, it's no longer BEIS, but as its successor, as the central body responsible for coordination of industrial and public sector assets. And then you talk there about maintaining the relationship with the UK Vaccines Industrial Base and HMG. Over the page 2, please, just a couple more aspects of this. We can see it says it would also monitor and relay current threat assessment of novel diseases to industrial partners, working in partnership with cross-government functions on horizon scanning and intelligence. And governance, please, a bit further down. You say an independent industrial experience chairman and board should be established to bring together the work of the various strands. So that is what you're setting out in that recommendation document. Thank you, that can come down. Before you expand on some of these points, perhaps, Dr. Dix, can I just put the counter-argument to you? Is it really necessary to add another permanent level of bureaucracy to the vaccine development landscape? Why can't we rely on existing systems that are in place now, and then, if necessary, just set up a VTF-like body when the next pandemic hits?
You can, and then you're relying on what the UK has at the time, which wasn't very much when we started the VTF, and probably wouldn't be, might be even less next time. But I think the real point here is this is part of peacetime now. The vaccine agent, if you think about vaccines are the best preventative medicine. We're talking about health prevention rather than waiting to deal with health problems. So there's a need to understand vaccines in a much broader context all the time, and to be sure that we as a country are vaccinating our population with the best vaccines for the diseases that are there already, and maybe ones that are coming down the line that aren't going to be pandemic or endemic. So there's a proper long-term role for a group that really understand this, and at the same time become world-renowned for being able to do it and understand it, and then link to all these bodies like the WHO, like CEPI, like all the, and be a world leader on the world. Name the person in the government that's a world leader in vaccines. You need it. And I think an agency might be the wrong word. I mean, I think it comes up with all sorts of connotations. It just needs that the UK needs a central body that understands vaccine and actually understands new vaccine developments that are happening and whether they're the right things to do, and encourage them to be done in the UK. And so we're really on top of it, and we are the world leader in the whole area of vaccines. Then you're sort of safe for the pandemic, because you've got so much access to everything and the knowledge.
If Matt Hancock was here now, he may say that such an agency as you've sketched it out was unnecessary, because that is what UK HSA are doing. That's essentially what he said when he was giving evidence to the inquiry a little earlier in this process. And we heard from Professor Dame Jenny Harries about the Vaccine Development and Evaluation Centre, or VDEC. Why aren't the systems that are in place now, put in place by the UK HSA, why aren't they sufficient in your view?
So first, I'd like to say that the UK HSA has a much broader remit and has to do an awful lot of things, and it does an awful lot of things very well for the country. That's important. But it hasn't got any leadership in vaccines. And the Vaccine Development Evaluation Centre, as it's called, was a small little lab when we started the VTF, with some very good people that could test vaccines if they were brought to them in live assays, and some very specialist stuff. That's what it is. It's a testing centre. It has no vision, it has no leadership, and no understanding of how you develop vaccines. So the name is false. It's actually just a laboratory testing centre, but a very specialised one. And I think that's misguided, or it misinforms, I would say.
The final thing I want to ask you about is this, that the inquiry has heard a suggestion from Lord Sharma that short of creating a national vaccines agency, what should be established is a vaccine expert advisory panel, a body which comprises industry experts, ministers, civil servants, perhaps people from academia, drawing together those strands, which is something you've stressed. They would meet regularly, horizon scan, audit the UK's capabilities, and make recommendations for investment. Rather than an agency, an expert panel, and then use that as the platform for establishing the VTF if and when a next pandemic hits. What are your thoughts on that?
I just don't think it'll work. I think you have to build, the agency has to be a place that, or the activity has to be a place that fosters the relationships with industry. An advisory panel getting together every so often is no way of doing that, and it has no power, so it's not going to make things happen. And we know what happens to advice. It gets put on the shelf. It doesn't get. It's got to be part of the business. It's got to be part of running the country, and so I just don't agree with advisory panels.
My lady, those are all.
Can I just pursue that point about an agency? As you'll probably know, it's not that easy to persuade any government, whatever political hue, to create a new body that'll cost money. How would you, if I were to be persuaded to make the recommendation, how would you frame it to persuade government that it was worth the amount of money it would cost to set up the agency in the long term?
I think I would use the analogy of the MOD. And we spend a lot of money there, and we have real expert people. We have military soldiers that know what they're doing, running it and being part of it. And unless we have that in the UK for vaccines, then when the next pandemic comes, we'll be back where we started. We really need to invest in, and I think investing in vaccines for health is the way to think about it, not for a long-term future thing. That's very hard to justify, but knowing that having it for health and being the best place in the world for vaccines means that you are covered. And I don't think it needs to be hugely expensive either. It's not they won't be doing. They will actually be corralling and bringing on board the industry and making it all work and helping putting it together. So I don't think it needs to be this huge cost centre.
Is there any argument that if such an agency was set up and the expertise were gathered together and they corralled and developed the relationships you're talking about, that might attract investment into the UK?
Absolutely. I think
that's the kind of argument that might appeal to government.
Well, I think that's what I was saying earlier about the VTF actually leveraged the activities in the UK to attract investment. So we were very close with 2 other companies of having them come to the UK and set up their manufacturing here as I left. And I understand that those discussions just became so entrenched in government thinking that they just walked away in the end because they weren't being given the vision of why it'd be great to be here. And there's plenty of very good things the UK has that you can actually attract companies to come to the UK.
Thank you. Just a couple more questions to Mr. Thomas, who's over there.
Good afternoon, Dr. Dix. I've just got a couple of questions for you. My name's Leslie Thomas. I'm representing FEMHO, the Federation of Ethnic Minority Healthcare Organisations. At paragraph 7.4 of your statement, you make mention of the communication plan that was fronted by Dame Kate Bingham. You state that our members of the Vaccine Taskforce were asked to participate in podcasts and Zoom meetings with various ethnic groups to help disseminate understanding of vaccines. As I recall, this was frowned upon by HMG Comms, that's the Government Communication Centre, and eventually stopped. In particular, the Vaccine Taskforce podcast had been paused indefinitely by the 17th of November 2020, with one episode remaining unpublished. In my opinion, this was wholly short-sighted. Dr. Dix, could you elaborate on the reasons why the Vaccine Taskforce sought to participate in the podcasts and Zoom meetings with ethnic groups? Why was that important?
We felt that as a group we had a level of credibility and trust, and that these groups desperately needed information, and they needed to be given solid information that was well thought through and presented in a way that was digestible by different groups.
Okay, just on that, what sort of reach were you expecting to have?
Well, you never know with podcasts. Sometimes they go nowhere, and sometimes they become the podcast of the year. So we were working with communicators to actually make those podcasts, live and real. So we didn't really know what the reach. We just thought it was an important thing to do.
And further, at paragraph 7.5 of your statement, you say this. You say, HMG Comms did not want the Vaccine Taskforce to participate in comms. In fact, they were paranoid about the Vaccine Taskforce saying anything publicly. Question. Why did you think this type of communication was frowned upon by HMG Comms?
I don't really know, apart from that the government was fixated in controlling all information, and there was probably a lack of trust, I think. But we were just told to stop doing it, or Kate was told to stop doing it, and she had a proper programme of communication, and it was told that it was the government's job to do that and not the task force.
In any event, no rational explanation provided?
No, there wasn't a rational reason. It was paranoia, I think.
Thank you, my lady.
Thank you, Mr. Thomas. Dr. Dix, I'm going to embarrass you again. To be described as phenomenal by someone like Kate Bingham is praise indeed. So
I had to pay her a lot of money.
I'm sure you did.
No.
But we're very fortunate that people with your skills and ability and experience were prepared to step up in a national emergency. So on behalf of us all, I express my gratitude for what you did during the pandemic and for your help to the inquiry.
Thank you, and I'd do it again.
Good.
Thank you.
Right, I think that completes the evidence, Mr. Mason.
It does, my lady.
Very well. I shall return at 1:45 to begin the closing submissions.
All right.